Hello, thank you for joining. My name is Pavel Princev, and I am a Director of Business Development at Bridge Biotherapeutics. As a public company, the usual disclaimer is applied, and my remarks today may contain forward-looking statements. In this presentation, I will briefly introduce to you our company, as well as give a short introduction to our 3 clinical stage programs. Bridge Biotherapeutics is a clinical stage biotech company founded in 2015 with its headquarters located in South Korea and with additional offices and labs spread out across the United States and China. The South Korean branch oversees the clinical stage research and development of our core pipeline, sets the overall company business strategy, and leads BD initiatives that are focused primarily on the Asia-Pacific region. The Boston Discovery Center, otherwise referred to as the BDC, was established in October of last year and is located in Cambridge, Massachusetts. The BDC, being located across the Pacific, oversees new business development across North America and Europe, while also carrying out R&D work for their own discovery programs. The goal of the BDC is to initiate discovery programs based on selecting innovative targets and understanding novel biology with an added focus on translational strategy to address highly unmet medical needs. We believe that our bicontinental strategy not only enables us to have a highly efficient global workflow, but also places us directly in key regions that are most well known for cultivating highly impactful academic research, as well as innovative and cutting-edge scientific discovery found from the startup level all the way up to big pharma. Equally important is positioning ourselves in these locations allows us to assemble a team of the most talented and skilled industry experts across their respective nations. We have 7 people in our senior management team. James Lee, the founder and CEO of Bridge Biotherapeutics, comes from a background in structural biology from Seoul National University, who has over 25 years of industry experience in research planning, and business development at LG Chem. Prior to founding Bridge Biotherapeutics, James has also founded and co-founded 2 other biotech companies in South Korea. Along with James, our well-balanced management team is comprised of experts spanning a range of disciplines, including scientists, entrepreneurs, financiers, and medical doctors, to name a few. Within the first few years after our foundation in 2015, Bridge Biotherapeutics in-licensed several preclinical stage drug candidates from various domestic research institutes and partners to build its internal pipeline. With our small team of industry experts, we further developed the assets until they received IND approvals, upon which we brought them into clinical testing, where they are now. During this time, we also formed several other collaborative research partnerships to strengthen and expand our discovery research initiatives. The success of our discovery external and develop internal strategy helped form a strong and stable foundation that enabled us to take our company public in 2019. From this year, we have entered our growth phase. The Boston Discovery Center is now fully operational with their own discovery pipeline. Within this year alone, we have increased our headcount by 25%. And by the end of 2025, we aim to surpass 100 employees in our workforce globally. Additionally, we strive to increase our clinical pipeline from 3 to 7 programs through our active global BD in-licensing efforts. And within the next 4 years, we hope to have our first FDA marketing approved drug. Finally, for the evolution phase and beyond, we envision to transform Bridge Biotherapeutics into a global stage company with an established sales network in the US with over 500 employees worldwide. Since January 2020, we have substantially expanded our workforce across the board. In order to keep up with the pace of our developing research projects, we have doubled the size of the R&D team. Similarly, we have also increased our BD workforce so that there is more personnel available to focus specifically on domestic and ex-Korea partnering, as well as our global search and evaluation activities. The strategy and management team has also been expanded considerably over the past 2 years. Currently, we have 3 clinical stage programs under development. BBT-401 is the most advanced, which is a first-in-class polyno-1 inhibitor for the treatment of ulcerative colitis that is currently in phase 2. In December of 2018, we formed a strategic alliance with Daewoong Pharmaceuticals for the development and commercialization of BBT-401 in 22 countries across Asia, including China. Both of our companies are expected to further accelerate business development activities in the Asian territories, where there is a heightened demand for an effective ulcerative colitis therapy. BBT-877 is a first-in-class autotaxin inhibitor for the treatment of fibrotic diseases such as idiopathic pulmonary fibrosis. The phase 1 trial is complete, where we demonstrated good clinical safety and produced strong biomarker results. And we are now planning the phase 2 trial design and hope to initiate the study sometime next year. And our third program, BBT-176, is a first-in-class fourth-generation EGFR tyrosine kinase inhibitor with potent and selective activity against a broad range of emerging acquired C797S mutations that are prevalent in EGFR-positive non-small cell lung cancer. We recently initiated the phase 1 dose escalation trial across 3 sites in South Korea and had our first patient dosed in April of this year. Starting off with BBT-401, we have developed a first-in-class polyno-1 inhibitor for the treatment of ulcerative colitis. BBT-401 inhibits polyno-1 via a novel mechanism of action and has shown to have strong safety and anti-inflammatory efficacy profile in our preclinical studies, eventually paving the way for its entrance into the clinic. Given that the location of UC-affected tissue is deep in the GI tract, BBT-401 has been developed as an oral formulation with a gut-restricted distribution profile that minimizes systemic exposure and increases site-specific proximity of the drug. With this formulation, we have obtained an excellent phase 1 clinical safety profile and are now evaluating the efficacy of the drug in the phase 2A trial. For the phase 2A trial, the low-dose cohort has been completed, and we are now currently enrolling patients in the mid to high-dose cohorts across several sites in Korea, the US, New Zealand, and Eastern Europe. The left-hand side graphic illustrates the novel mechanism of action of BBT-401. It is well known that toll-like receptor signaling plays a key role in the pathogenesis of several inflammatory diseases, with UC being one of them. BBT-401 is a protein-protein inhibitor that disrupts this aberrant signaling pathway by preventing the formation of the polyno-1 containing MyD88 and RIP1 complexes. By doing so, downstream NF-kappa beta is unable to translocate into the nucleus and initiate the induction of pro-inflammatory genes that lead to the formation and exacerbation of ulcerative colitis. In the middle preclinical efficacy chart, BBT-401 was administered to mice with DSS-induced colitis at 3 dosages, low, mid, and high. We were able to see a clear dose-dependent effect across the 3 dosage levels. And when compared to a competitor, the medium and high dosage groups showed a significant reduction in disease severity, which is determined as a cumulative score that takes into account the extent of induced anti-inflammatory and mucosal generation effects of the drug. On the right-hand side table is some key clinical efficacy data of our low-dose cohort from the phase 2A trial. Over an 8-week period, patients were given our gut-restrictive oral formulation of BBT-401, upon which drug efficacy was measured using the industry-standard Mayo score. Our drug has shown to reduce the Mayo score in several of the treated patient groups, as well as reduce the presence of a key disease biomarker when compared to control. With the low-dose cohort portion of the phase 2A trial completed, we are now dosing patients in the medium to high dose cohorts. This is being conducted globally with trial sites in the US, Korea, New Zealand, and in Eastern Europe. And just last month, we have initiated the phase 1 China ethno-bridging study in healthy volunteers, which we expect to be completed by the first quarter of next year. Our next clinical asset is BBT-877, a first-in-class autotaxin inhibitor for treating IPF and other fibrotic diseases. Idiopathic pulmonary fibrosis is the most common form of idiopathic interstitial lung disease. It is a devastating lung disease that progressively worsens over time. The disease induces scarring and inflammation in the lung, interfering with the patient's ability to absorb oxygen from the air. The current standard of care varies with the severity of the disease. However, treatment options typically include supplemental oxygen, symptom management, lifestyle changes, and in some cases, treatment with antifibrotics. The existing treatment options, however, are still largely inadequate, with about 70% of patients dying within the first 5 years of diagnosis. BBT-877 has successfully completed its phase 1 studies, and we are currently in discussions with the FDA for planning the initiation of the phase 2 trial. Autotaxin is an enzyme that has been found in higher concentrations in patients with inflammatory and fibrotic diseases, as well as in tissues of patients with fibrosis of the lung. Autotaxin catalyzes both the hydrolysis of LPC and the local production of LPA. In turn, that LPA then binds with and activates several LPA receptors, triggering a downstream signaling cascade that leads to epithelial apoptosis, the primary initiating pathogenic event in modeled pulmonary fibrosis and IPF. Furthermore, LPA has also been associated with promoting inflammation and pulmonary edema, as well as other pathogenic events that further exacerbate the disease. The selective binding of BBT877 to autotaxin effectively inhibits autotaxin's ability to catalyze LPC and promote LPA production, greatly slowing down the progression of the disease. The chart on the right displays the LPA levels in patients receiving BBT877 in a multiple ascending dose from our phase 1 trial. It can be seen that for 800 milligrams QD dosing, a greater than 80% LPA inhibition was observed at steady state levels when compared to baseline. For BID dosing, 80% and 90% LPA inhibition levels were achieved at the 100 and 200 milligram dosages respectively. With the phase 1 trial having been completed last year, we are currently in discussions with the FDA regarding our phase 2 trial design and plan. A follow-up meeting with the FDA is scheduled for early next year, where we hope to begin the phase 2 trial for BBT877 sometime in early to mid-2022. Our final clinical stage asset is BBT176. It is a first-in-class, fourth-generation EGFR TKI for the treatment of non-small cell lung cancer. NSCLC is the largest subset of lung cancer that accounts for 85% of all lung cancer cases and is also the most common cancer with the highest death toll worldwide. Due to the aggressive nature of non-small cell lung cancer, the average 5-year survival after diagnosis is often below 5%. One of the most well-known drivers of NSCLC are mutations in the EGFR gene. Previous generation EGFR TKIs target such mutations and have shown promising results, particularly the third-generation EGFR TKI blockbuster drug osimertinib. However, the majority of patients taking osimertinib eventually stop responding to it because they have developed new mutations in EGFR, which prevents the drug from working properly. This key resistance mutation has been identified as C797S and greatly reduces the efficacy of third-generation EGFR TKIs. Patients with C797S mutations are left with limited treatment options, making the development of a fourth-generation EGFR TKI that is active against C797S mutations all the more important. BBT176 has shown outstanding preclinical safety and efficacy when compared to existing and developed products. Given its excellent preclinical profile, BBT176 has become the world's first fourth-generation EGFR TKI to enter the clinic, with the first-in-human dosing for our phase 1 dose escalation trial being completed in April of this year. Concurrently, we are also continuing to discover and develop additional non-small cell lung cancer treatment solutions that are effective in many situations, including C797S double mutations. This slide summarizes the biochemical and cellular activities of BBT176. We conducted a head-to-head comparison between our compound and the third-generation EGFR TKI osimertinib, as well as the first-generation gefitinib. As can be seen in the top panel, summarizing the cell-free biochemical assay data, BBT176 shows a broad and potent activity against single driver mutations, as well as the C797S containing double and triple EGFR mutations, all with IC50s in the single nanomolar range and sub-20 nanomolar range for the double mutations. Moving on to the cell-based assay data, we see that BBT176 remains consistent with what was seen in the biochemical assay. It is active in single, double, and triple mutations with heightened potency towards DEL19 containing mutations. In both assays, BBT176 shows weak activity against wild-type EGFR, demonstrating its specificity for EGFR mutants. While osimertinib displays activity against driver mutations in both assays, it has very poor activity against the C797S containing triple mutations. These are the primary mutations responsible for the acquired resistance to osimertinib and is where BBT176 excels. Here is a summary of our in vivo efficacy experiments. We tested BBT176 across a wide range of cell and patient-derived xenograft models, as well as a brain metastasis model, all of which showed either clear tumor regression or inhibition. In this experiment, we were interested to see how BBT-176 responds in a PDX model of a patient who has been through 7 lines of therapy, including chemotherapy, erlotinib, osimertinib, and has been found to carry a DTC triple mutation. We were able to see a dose-dependent anti-tumor effect with tumor regression achieved at 100 mpk administered once daily, all while having a minimal effect on body weight change. Right now, the Part 1 dose escalation trial is underway across 3 sites in South Korea. We recently completed cohort number 3 and will soon begin dosing patients in the 4th cohort. Since the EGFR mutation is more prevalent in the Asian demographic, we are also interested in comparing PK and safety between Asian and Caucasian patients, which we plan to do for our last cohort in the Part 1 study. The Part 2 dose expansion study will be evaluating the efficacy and safety of BBT-176 at the recommended phase 2 dose, focusing on patients that have confirmed C797 mutations. This trial is planned to be carried out in South Korea, the United States, and possibly other regions as well. We expect to initiate the Part 2 study towards the 4th quarter of next year. Our company has shown solid financials. These are some of the numbers at a glance. Our current market cap as of the end of September is $207 million with total assets valued at about $51 million. We also have a strong cash runway until the end of 2023. Our stock price has been somewhat less steady over the past year. However, we believe that our differentiated strategy, exploiting global science to address clear medical needs in combination with the potential milestones coming from our 3 early to mid-stage clinical studies, will start to get reflected in the broader market. To summarize, we have 1 multinational phase 2 study ongoing for ulcerative colitis therapy and an additional 2 phase 2 studies planned to begin next year for non-small cell lung cancer and idiopathic pulmonary fibrosis. All 3 of our clinical stage assets are first in class and are leading the way to bringing much needed medicine into the clinic, which will address substantial unmet needs in their respective indications. With the rapid growth of our company during the last year, we continue to diligently grow our internal R&D pipeline, both in Korea and in the United States, through new discovery efforts and open innovation, particularly from within the Boston Discovery Center. In the long term, we believe that Bridge Biotherapeutics is on track to becoming a fully integrated bicontinental biotech company. This concludes my presentation on Bridge Biotherapeutics. If you have any questions or would like to know more about any of our programs, please send us an email. We look forward to hearing from you. Thank you.